The topic of our lecture is organ of hearing and equilibrium. And this is not the very important topic among others, but it is very important to understand how do we perceive sounds and how to correct the progressive hearing loss with aging, and how to prevent, how to correct it. And the second purpose why to study this topic is to understand how do we maintain the balance of our body. How do we maintain the equilibrium, as problems with equilibrium, they accompany many neurological disorders. So let's get started. And first of all, we should say that there are three compartments of our ear, and I hope that from anatomy department you already know a lot about the external and middle ear. Let's just refresh some knowledge. First of all, external ear is represented by the ear auricle, which primary purpose is to collect sound waves. After that, external auditory canal or external acoustic meatus, which conducts those sound waves to the tympanic membrane. Exactly this membrane separates the middle ear cavity from the external ear. Then, vibration of the tympanic membrane is conducted further through three ossicles to the internal ear. And here in the middle ear cavity, also we can find a muscle, which is tensor tympani muscle. What's the function? The function is to regulate the tension of the tympanic membrane. So depending on the sound waves, if the sound is too loud, this muscle relaxes a little bit. So this mechanism prevents the damage of our internal ear by too loud sounds. So if sound is too loud, this muscle regulates the tension and decreases the tension of the tympanic membrane. So we can modify and modulate our hear perception depending on circumstances. Okay, notice that ossicles are fixed by ligaments to the cavity. And in histology department we do not discuss, but from anatomy department you know a lot about the structures surrounding this cavity, and it is important. Notice that there is a tube which is called eustachian or auditory tube, and it communicates the middle ear cavity with the nasopharynx. So it is lined with the same epithelial tissue, which is pseudostratified ciliated. What's the reason of having this tube? I think that you travel a lot with the help of by plane, and there is always differences in the pressure once the plane ascends, and of course you know that people, they are recommended to chew something, to swallow something, or to yawn, just to equalize the pressure by each side of the tympanic membrane. As you can see, this membrane is exposed to the outer environment through the external auditory canal, and middle cavity should not be insulated. It also is communicated through the auditory canal, through the nasopharynx, but usually this communication is blocked, but when we swallow something, it gets opened. Okay, and doctors should remember about this tube because it is used by different infections to ascend. So if somebody, your patient, suffers from the throat ache or tonsillitis, any infection in the pharynx, in the mouth, it might ascend to the ear through this tube. That's why at the entrance of this tube there is usually tubal tonsil. Remember the pharyngeal lymphoid ring, it includes sometimes tubal tonsils. Okay, this is as for the middle ear cavity, and let's move on. Yeah, tympanic membrane, several words, very important membrane, and it is covered by two different epithelial tissues from the external surface and from the side of the middle ear cavity. So skin epidermis like epithelial tissue lines the outer surface, and in the middle ear the same epithelial tissue which lines the cavity. And between those two there is a core of connective tissue with collagen fibers inside. They provide the tension, and we can distinguish the so-called pars tensa of the tympanic membrane and pars flaccida. Here is it, and we can see here the malleus, the first ossicle, which is in contact with the tympanic membrane. Okay, and now we are to investigate in details the structure of the internal ear. It really resembles the snail with its shell and a soft snail inside them. So the shell is represented by the bony labyrinth. Bony labyrinth is a system of cavities inside the temporal bone. Those cavities are filled with a perilymph. Perilymph resembles extracellular fluid, which is rich in the sodium ions, as usual extracellular fluids. And then it includes three semicircular ducts, canals, three semicircular canals, here they are, then the vestibule, here is it, and the cochlea, this red one, spirally oriented. Then inside this bony labyrinth, inside those cavities, inside the perilymph, here there is a membranous labyrinth, which is filled with the endolymph. Endolymph is called so because it resembles the intracellular fluid, rich in potassium ions. That's why it is endolymph, like intracellular fluid. And here we can see inside semicircular canals there are semicircular ducts put inside. Then inside the vestibule there is a saccule and utricle, two cavities. Not very exactly seen, but there are two cavities, spherical cavities. And inside the cochlea there is a cochlear duct. So like inside the shell there is a snail, inside the cochlea there is a cochlear duct. And the same is true for other compartments. And this soft membranous labyrinth bathes inside bony labyrinth, inside its perilymph. Notice that membranous labyrinth is divided in a vestibular part, which is responsible for the equilibrium, and cochlear part, which is responsible for our hear perception. Okay, the same is shown here. Let's not stop at this. And here, this is the key slide for the whole lecture. And before understanding all the other information, first we should capture this. So this table concentrates the general information about every compartment of the internal ear. So first of all, we should comprehend that inside semicircular ducts, in their dilated portions, there are so-called Crista ampullaris. These are the receptor apparatus destined to perceive angular acceleration, rotational movement in other words. So exactly semicircular ducts, they help us to understand the rotational movements. If we close the eye but rotating our body, we still understand that we are rotating in the horizontal plane, for example, leftwards. That's because of the semicircular ducts, because of the Crista ampullaris, and that's what we call perception of angular acceleration. You may say rotational movement in other words. Okay. Then in the vestibule, in our utricle and saccule, there are macula. Macula is in a special receptor apparatus destined to perceive linear movements, gravitation and vibration. Linear acceleration means movement along the line. Gravitation and vibration. So once you are in the lift and you understand whether you are moving upwards or downwards because of the macula. And today we are to understand how it works. Okay, so semicircular ducts are needed to perceive rotational movement. Vestibule with its utricle and saccule, with the help of macula, they perceive linear movements. And our cochlea, at last, with the help of the organ of Corti, perceives sound waves. And now let's investigate each compartment one by one and each receptor apparatus in details. And we start from the cochlea and hear sound perception. Cochlea is called so because really it resembles a shell with 2.5 turns around the axis. The axis is called modulus. Modulus is in the center. So the walls of this shell build-up of bone. This is the system of cavities in the temporal bone. Then, if we make a cross-section, we'll find out that inside the bony labyrinth, there is a membranous labyrinth, which is of red color. So, membranous labyrinth, it is fixed in the middle of the bony one, and membranous labyrinth divides bony into two parts. The upper one, which is called scala vestibuli, and the lower one, which is called scala tympani. And both of them are filled with a perilymph, as bony labyrinth does. And the middle one, the membranous labyrinth, which is called cochlear duct, also it is called scala media, it is filled with the endolymph. Remember, again, endolymph has high potassium concentration as inside the cell. Perilymph has high sodium concentration as outside the cell. Good. Then let's continue. What is especially important to understand in this slide is that scala means stairs, but spiral stairs. Scala highlights that each next turn is located a little bit higher comparing with the previous one. So if we enter the cochlea and move along its duct, then with time we ascend, like if we move along the spiral stairs. That's why each compartment is called scala. Something like there are 3 spiral stairs, one above another, and they ascend to the top. So the next aspect, very important, is that at the top of the cochlea, at the most apical part, there is a communication between the scala vestibuli and scala tympani. So if we enter the cochlea through the scala vestibuli and ascend on its top, at the end there is a door, and opening this door we descend from the scala vestibuli to the scala tympani, and through another stair system we descend back. Okay, and there is one more aspect we should comprehend about the structure of the cochlea. So cochlea is communicated with the middle ear cavity, and there are 2 openings. The first one is directly blocked with the stapes, the 3rd ossicle, and it is called oval window, and it starts and it opens to the scala vestibuli. So scala vestibuli at the base starts from the oval window, which is in communication with the stapes, and scala tympani also is in communication with the middle ear, and it is closed with a round window, which is closed with a secondary tympanic membrane. Let's pretend back here. Yeah, and that's what is shown. So middle cavity, middle ear cavity, here we can see 3 ossicles. So the 3rd ossicle, it is in contact with the oval window, and exactly from oval window scala vestibuli starts, while scala tympani starts from the round window, which is blocked with a secondary tympanic membrane. Okay, so this is the general information about the cochlea. Now let's investigate it in details. So if we look precisely at the membranous labyrinth and the scala media, the cochlear duct, we'll find out that on the lateral side there is a special stripe of epithelial tissue which is called stria vascularis. And this is a very unusual site where epithelial tissue contains blood vessels. As you know, the general feature of epithelial tissues is that they are devoid of blood vessels. But here in this place, they have a very rich network of capillaries, and their main function is to produce the endolymph and to regulate the ion balance here. Once again, I remind that this liquid has high potassium concentration, and in this it resembles the intracellular fluid, and exactly the stria vascularis is responsible for producing it. Okay, then we move forwards, And the second aspect we have to discuss in the cochlea is the receptor apparatus, which is the organ of Corti or the spiral organ. Exactly here receptor cells are located, exactly here sound perception takes place. So let's investigate its cellular composition, and after that we'll discuss how they perceive sounds. So here the structure is shown. If we make a cross section, we'll find out that in the scala media on the basilar membrane, which contains many collagen fibers, there is a group of cells which could be divided into receptor cells, which are yellow, and supporting cells. In general, two groups are considered. Then notice that among receptor cells we can distinguish pillar cells. They are called so because they are tall as pillars, and pillar cells, they form internal tunnel, or the tunnel of Corti. This inner tunnel divides all the cells into two groups. By one side, close to the modulus, to the central axis, there are internal cells. By the another hand, there are external cells close to the stria vascularis. And notice that external cells are more numerous than internal one. Okay, so cells are divided into receptor and supporting, first of all, and all those cells, both receptor and supporting, are divided into internal and external, depending on their position according to the inner tunnel. Okay, now, receptor cells, they are called hair cells. Why hair? Because on the apical side, they have long microvilli called stereocilia, and they really looks like hairs, and that's why they are called hair cells. And exactly with the help of stereocilia, they perceive sound waves. So receptor cells, hair cells with long stereocilia on the apex. Then what about supporting cells? We have already mentioned pillar cells. They form this tunnel. Then the second type, extremely important, these are the Deiter cells. Another name is Phalangeal cells. Names will be written in another slide, so now we just discuss. Phalangeal cells, as you see, they hold receptor cells, so they mechanically support them. And the second function is that they send a long phalanx, forming an umbrella-like covering at the top of the hair cells, properly insulating bodies of receptor cells from the stereocilium. Okay, so phalangeal cells, they insulate the Corti organ from the endolymph, because in the scala media there is an endolymph and there is a high potassium concentration, while inside the organ of Corti the same perilymph as in the tympanic scala tympani, because through the basilar membrane it percolates and washes all the cells from outside. So cells, they are located in the typical extracellular fluid which percolates from the scala tympani, but stereocilia are exposed to the endolymph in the scala medium. It's very important. We'll return to that later. Also, there is a system of cells of Hansen, Batcher, Claudius. Not very important. There is no special function. I don't ask you to remember about that. So at least remember pillar, phalangeal, and hair cells. Okay, and one more element should be added for the complete understanding. This is the tictorial membrane, jelly-like covering, which is in contact especially with the outer hair cells. Okay. Notice that there is one row of the inner hair cells and 3 rows of the outer hair cells. Now let's continue. Then those cells are written here. So there are 2 types. Once again, receptor cells, they are hair cells, here they are, and supporting cells, they are green one, and supporting cells, Deiters, here they are, then pillar cells and Hansen, Claudius, Becher, not so important as others. And let's pay attention to the hair cells, as they are the most crucial in the generation of action potential. Hair receptors. They're equipped with a long stereocilia, and here they are shown with the help of the scanning electron microscope. So there are 3 rows of outer hair cells and 1 row of the inner. Notice that inside the stereocilia there is a bundle of actin, which is cross-linked with the help of asparin and fimbrine proteins. And these cross-links are very important, as stereocilia should be linear. It should not coil, and it should be exactly rigid. Because mutations in those proteins, they result in the formation of soft stereocilia, and it can't perform its function. To function properly, it should be rigid, linear, and cross links are very important. Then, on the surface of its membrane, exactly in the stereocilium, there is a special mechanoelectric transducer channel protein, which helps to open the channels once the membrane is stretched. So exactly this transducer transforms mechanical energy into electric nerve impulse. Now let's discuss how it works, but before doing that, we should say that there are 2 types of hair cells, as you remember, inner and outer, and we call them type 1 and type 2. So they differ not only by location, but also by the structure and function. Notice that type 1 cells, they are inner, and they are flask-like, so they have the more dilated base, and they are surrounded, what is especially important, with a large chalice of afferent nerve ending. Afferent means the information is conducted from the hair cell to the brain. To the brain. What about the second type? Second type differs by the shape, as it is more columnar-like, cylinder-like, and it has afferent as well as efferent nerve endings at the base, and usually multiple nerve endings. That's the principal difference. And the question is, what is the reason of having efferent nerve fibers? We can understand that information about sound is brought to the brain, but what's the reason of opposite direction, opposite conduction? The general essence is to modulate the impulse transduction from the hair cells to the brain, to modulate, to facilitate, or instead to block in case of multiple sounds, or in case the sound is too loud, for example. So the efferent nerve endings, they have modulatory function, like the tensor tympani muscle helps to regulate the tension of the tympanic membrane. As well, efferent fibers, they modulate the sound perception on the level of hair cells, because it is very important to protect them, to prevent from damage by too loud sounds. Okay, so now we know almost everything, and the last detail is that stereocilia are connected one by one with the help of tip links. Those tip links, they stretch the membrane in case of deflection. So imagine stereocilia are deflected, tip links, they stretch, and this stretching results in the opening of ion channels. And in this way, mechanical energy, mechanical stretching, results in the opening. That's why hair cells, they are equipped with mechanosensitive ion channels. Let's look how it works. Once again, there is another diagram showing us 2 types, and in text the information is given, we have just discussed. Okay, besides here, diagram shows that this is the scanning electron micrograph showing us 3 outer hair cells. You see, they are of which type? What do you think? Of course, they are of type 2, because they are cylindrical in shape. That's what we can distinguish. And you remember they have afferent as well as efferent nerve endings. Notice besides, despite outer cells, outer hair cells or type 2 hair cells are more numerous, 90% of all the information we get from inner hair cells, okay, type 1 cells, the paradox. Okay, and now let's discuss in details the function of phalangeal cells or Deiter cells. Now we know a lot about the hair cells, and as there is a nerve ending at their base, so synaptic connection should be formed, those cells, they can't be fixed to the basal membrane themselves. And exactly phalangeal cell helps to hold the hair cell and helps to form proper synaptic connections. So the primary function of phalangeal cell is to provide the mechanical support. So there is something like holding in their arms hair cells, and which is also very important, as we have already said, they send a long phalangeal process, like phalange, and at the end an umbrella-like dilation is formed, which is connected by tight junctions with the same processes by the neighboring cells, and all together they create a continuous cuticular plate, properly insulating the medium around the cellular bodies and the medium in which the stereocilia base. Once again, stereocilia, they are put in the endolymph where potassium prevails, while cellular bodies of both hair phalangeal cells, they base in the perilymph, typical extracellular fluid. Okay, that's another important function of the phalangeal cells. Notice this pretty nice electron scanning electron micrograph showing us the phalangeal cell with its process and the hair cell. Here it is. And stereocilia of hair cells, they are exposed outside to the endolymph. Okay, now we know almost everything to understand the mechanism of sound perception. And here once again those cells are given. Let's skip it. Let's return to these slides later. So mechanism of sound perception. When the sound waves are collected by the ear auricle, they result in the oscillation of the tympanic membrane. Oscillation of the membrane. Let's return besides to the slide at the beginning, which is the most, for example here. So sound waves, they travel through the external auditory canal. They result in the vibration of the tympanic membrane. Its vibrations further are accelerated by a system of 3 ossicles, malleus, incus, and stapes, and as a result they cause the vibration of the oval window. You remember that oval window is at the beginning of the scala vestibuli. So vibration of the oval window results in the vibration of the perilymph at the base of the cochlea, and these vibrations, they propagate up to the apical portion of the cochlea, and by moving upwards, those vibrations, they can't displace the bony labyrinth as it is stiff and rigid, but instead they result in the vibration of the membranous labyrinth, which is soft and it is fixed to the bony. So vibration waves, they result in the vibration of the organ of Corti, which is located inside the scala media, inside the membranous labyrinth. Okay, so it is very important. So oval window, it generates the wave of perilymph in the scala vestibuli, and those waves, they ascend along the scala vestibuli upwards, and along their propagation they result in the vibration of the scala media as it is soft, and this is the part of membranous labyrinth. So what happens with the hair cells along with this vibration? Once the organ of Corti vibrates, hair cells, they start to vibrate as well. And what happens with their stereocilia? As they base in the liquid, due to displacement of the hair cells, stereocilia start to deflect. And once it is deflected properly, tip limbs, they get stretched, and potassium channels, they get opened. Entrance of potassium results in the depolarization of the cell, and as a result, calcium voltage gated channels get opened, calcium enters, and as a result, exocytosis of neurotransmitter takes place. Action potential is generated. So, what's unusual in this case? Unusual is that instead of usually sodium enters to depolarize, in this case, potassium enters. Why? The reason is, typical reason is unknown, but here I put the information for the most curious students. What is written here, that the membrane potential in the stereocilia and cellular body of the hair cell, they differ significantly. And that's why potassium enters the stereocilia despite its concentration in the cell is high. And as a result, potassium depolarizes the membrane as well as repolarizes it. And that's how it works. So the most important aspect we have to comprehend is that sound waves, they transform into the waves of perilymph, and perilymph results in the vibration of the membranous labyrinth together with the organ of Corti. Vibration of the organ of Corti results in the deflection of stereocilia, and this is exactly what is used to open the potassium channels. Okay, and now we should say that once we hear the sound wave, once we hear the sound, not all hair cells are excited at once, but only a small segment of cochlea works. That's because we perceive sounds of different intensity, of different frequency. And in case of high frequency sounds, they are perceived at the apex of the cochlea. Only here hair cells generate the action potential. In case of high frequency sounds, high frequency sounds are perceived at the base of the cochlea, close to the oval, to the round window. And in case of high frequency, only here hair cells work. That's what we call tonotopic organization. And it really resembles the piano, because in a piano, once you press any key, you generate exact sound of exact frequency. And in our ear, the opposite process takes place. The sound of this particular frequency results in the activation of hair cells, but in an exact place along this duct. So we have spirally oriented organ of Corti and many hair cells along its length, because not all of them are working simultaneously, but only a group of them. And it really resembles the spirally oriented piano, but this piano is used not to generate sounds, but to generate nerve impulses. Like opposite process takes place, but the same principle is at the base. Okay, then what should I say here, please? This QR code is given to recommend you to watch a special video. This video explains perfectly the mechanism of how do we perceive sounds, and I recommend you, this is my strong recommendation, and this is the best video not only about the hearing but among all the histology, very perfectly explaining. Please watch it and you'll understand everything at once, and watch till the end because the internal ear is shown at the end. Okay, so tonotopic organization means that sounds of different frequencies are perceived by different groups of hair cells, and it is important to take into account. Let's now return to the structure of the organ of Corti in the specimen. I remind you that at the end of the discipline in winter, you are going to have exam not only with the conventional MCQs but also specimens will be used, so you should deal with specimens. Let's try to investigate it. It is not very difficult. This is the cross-section through the cochlea. So what we can find here, this is the bony labyrinth. This is the bone, and exactly here the perilymph is, here and there. So this in the middle, triangular in shape, this is the cross-section through the membranous labyrinth or the cochlear duct, or the scala media, which is called with the endolymph. And here exactly high potassium concentration. This endolymph is produced by the stria vascularis. Here is it. And you may see there are lots of blood vessels inside. Then a spiral ligament is used to attach the scala media or the cochlear duct to the bony labyrinth. So here is the bony labyrinth, and here is the spiral ligament, helps to fix scala media, cochlear duct, membranous labyrinth to the bony one. Okay, so membranous labyrinth divides the bony labyrinth into scala vestibuli and scala tympani. There is a vestibular membrane or Reisner's membrane which divides scala vestibuli from scala media, and there is another basilar membrane bordering the scala media membranous labyrinth from the scala tympani. Okay, then in the center of the cochlea there is a medullus or the central axis, and inside there is a spiral ganglion. Spiral ganglion contains the second neuron in the chain, and exactly here neurons from the hair cells are located, and then they send the impulse to the cochlear nerve, and this is the part of the 8th craniofacial nerves. Okay, so what else can we distinguish here? This is the organ of Corti. Notice this is the tectorial membrane, jelly-like covering. Besides, when the scala media vibrates, tectorial membrane, it slides along the surface of hair cells, also stimulating the deflection of stereocilia. So tectorial membrane participates in the nerve impulse generation. And next slide gives us a higher magnification of the organ of Corti. So tectorial membrane is here. Pillar cells, we can find the outer and the inner one. Inside there is an inner tunnel. Then we can see exactly the outer hair cells. These are the 3 rows of nuclei, and at the base 3 outer phalangeal cells. At the base exactly one phalangeal corresponds to 1 hair cell. There are another additional supporting cells I don't ask you to remember, and here is the inner hair cell, and inner phalangeal is not very well distinguishable, should be right here. And there is a basilar membrane with many collagen fibers inside. Those collagen fibers, they are called strings, and depending on their length and tension, different sounds, they vibrate different parts of the organ of Corti with different effect. So the most effective vibration, low frequency sounds, they make at the top of the cochlea, yeah. And high frequency sounds, they vibrate most effectively the basilar membrane at the base. That's because of collagen fibers, of those collagen strings, which differ by their length and tension. Okay, I think we have considered everything about the organ of hearing, about the perception of sounds. Please watch this video, very helpful really, if you want to understand, not to learn by heart. And now, after we have discussed the cochlear duct with its organ of Corti inside, after we have discussed the sound perception, let's move on and discuss how do we perceive the movement and the position of our body in a 3-dimensional space. First of all, we should say that there are the same hair cells, type 1 and type 2, in Crista, ampullaris or Macula. So receptor cells are the same. But what's the difference is that they are equipped not only with a stereocilia shown with blue color, but one long kinocilium. Kinocilium is present. Kinocilium is built up of microtubules. So kinocilium is a true cilium, while stereocilia are microvilli. Okay, this is the subject of eternal confusion, but just I'm sure you'll cope. So stereocilia, they contain actin filaments inside, while kinocilium, it contains 9 pairs of microtubules. And now the question is, what is the reason of having the kinocilium? What is the function? The function is that once the stereocilia are deflected towards the kinocilium, it results in the stretching of tip links and opening of potassium channels. But when opposite situation takes place, when kinocilium is deflected towards the stereocilium, tip links, they get relaxed and nothing happens. So cell get hyperpolarized and it doesn't generate the nerve impulse. So kinocilium provides polarity, and here the direction matters. And why direction matters? Because this receptor apparatus destined to perceive our movements, and if we move along the line, it is important also to understand we are moving forwards or backwards. And to reveal that, we need a kinocilium. And cells, they are located in different orientation. So once we are moving forwards, in one group of cells, stereocilia are deflected towards the kinocilium. So they get activated and they are informing we are moving forwards. While the opposite cells, they have their kinocilia deflected towards the stereocilia, so they are inactivated. Once we move backwards, the opposite situation is true. So always only a part of cells work, because some of them, they are informing about moving forward, some of them about movement backwards. And to detect the direction, kinocilium is important. So in the organ of Corti, in the organ of hearing, there is no kinocilium because there is no reason of detecting the direction of the sound, only the frequency, yeah? But in the organ of equilibrium, it is important not only to understand in which plane or along which line we are moving, but also to understand the direction. Okay, so let's first talk about the semicircular ducts. There are 3 of them, and their function. We remember from the beginning of the lecture that semicircular ducts are needed to perceive the rotational movement. And we can rotate in 3 different planes. So in the horizontal plane we can rotate, yeah, then in the sagittal plane, and also in a frontal plane. And to perceive all those types of movement, of rotation, we need 3 semicircular ducts. One is in the horizontal plane, one in the sagittal, and one is frontal. And in every duct, in one of those 3 ducts, we have a dilated portion, and inside this dilated portion, a fold of epithelial tissue forms a special receptor apparatus which is called Crista ampullaris. Crista ampullaris is longitudinal and it is located perpendicularly to the long axis of the duct, so it almost closes it. It is located perpendicular. And one more detail we need to know is that the duct is filled with the endolymph. Notice that bony labyrinth is filled with a perilymph. It is with blue color, high sodium, and Crista ampullaris bathe in the endolymph, where is high potassium concentration. Then, how it works, it is very important to understand, and let's imagine, and then we'll make one short experiment. So let's imagine a semicircular duct which is in the horizontal plane, and it is filled with the endolymph. So what happens once we rotate our head? The duct also rotates together with us. But what happens with the endolymph? Endolymph remains in the same position. So once we rotate, only walls of the duct rotate, but endolymph does not, by inertia. And how you can check it easily? Let's first explain till the end, and then we make an experiment. So, if we imagine a Crista ampullaris in this duct, imagine it is put in the liquid, it is washed by the endolymph, and it is located perpendicular to its axis. And it is fixed to the wall of the duct at its base. So what happens when we rotate our head? the semicircular duct rotates, but Crista ampullaris cannot because it is in the liquid. So once we rotate the head, it deflects in the opposite direction because it is stay in the water and water doesn't rotate. Okay, so our rotation always they result in the displacement of the Crista ampullaris in the opposite direction. So that's due to the inertia. Let's make an experiment. You will like it. There is a water, okay? I don't ask you to drink. So if you take a cup of water, of tea, a cup, a glass, doesn't matter. So if you pour water inside and then make this paper and write something on it. I wrote my name, for example, just to understand where is the top and whether it is rotated or not. Then if you put this paper on the surface of the water and then try to rotate the glass itself, and as a result you will see that it doesn't work because it get attached to the wall. So we should wait until it get wet and float freely. So it should flow freely. Okay, so and once you rotate the glass, now it works. So once you rotate, you see that the paper remains in the same position because water is not displaced. You displace only the walls of the glass. The same is true for the semicircular duct. So once we displace the duct and the lymph remains as water in this glass, it is not displaced. That's why Crista ampullaris get deflected, because its base moves together with the duct wall, while its apex, it floats freely in the liquid and it gets displaced in the opposite direction. So it is easy to understand, to imagine. If you got it, I'm very glad. Make an experiment. Do not drink. Okay, hope it was helpful. Let's continue. Yeah, besides, the same is true once we rotate our head in other positions, in the sagittal, in the frontal, so it deflects in the opposite direction because fluid is not displaced. That's why it is filled with the fluid, not with the water. And once Crista ampullaris get deflected, the stereocilia get stretched, potassium channels open, and the same is true as for hair cells in the organ of Corti. So hair cells are the same, the only one difference is the kinocilium, which is single, and it is needed to understand whether we are rotating leftwards or rightwards, because always a group of cell only is activated. Okay, so this mechanism is shown here, we've already discussed, and now the last aspect, how our macula works, and why do we need vestibule, and how linear movements are perceived. Two spherical cavities, saccule and utricle, are needed. In the utricle, the macula is oriented in a horizontal plane, and in the saccule, it is in the vertical plane. This receptor apparatus is called macula because here hair cells also are embedded in the jelly-like covering, but it is flattened, unlike cupula in the Crista ampullaris, and it is covered with sand-like, stone-like small stones called otoconia or otoliths. What does it mean? Oto means ear, lithos means stone, like ear stone, small ear stones. Calcium carbonate is at the base, and they make this membrane a little bit more heavy because inertia depends on the weight. And let's imagine what happens with the macula, which is put in a spherical cavity, once we move forwards. The same experiment again, but actually it doesn't work. If you take a bowl of water and if you move it along the line forwards, what happens with the water? It spills in the opposite direction. Yeah, because again, water has its inertia and it is not displaced with a bowl. And the same is true with the macula, as it is embedded in the endolymph. Once we move forward, macula is displaced backwards, and due to its displacement, it pull the hair of hair cells, and stretching of the hair results in the entrance of potassium. And polarization map showing us that cells, they are sitting not, they are oriented not in the same direction. So once we are moving forwards, one of them displays stereocilia towards the kinocilium, but another group in opposite direction. So once we move forwards, only a group of cells get excited. When we move backwards, they get hyperpolarized and active, but another group of cells activates, and that's because of the kinocilium. So the same is true for the Crista, ampullaris, and for the Macula. Okay, so let's summarize this information. Macula is represented by also two types of hair cells, type 1, type 2. Their stereocilia and kinocilia are embedded in the jelly-like otolithic membrane. It is called so because it is heavily covered with otoliths. Otoliths, carbonate crystal of calcium. And macula is washed by the endolymph as it is put in the membranous labyrinth. And once we are displaced forwards or backwards, maybe leftwards, rightwards, in this case, macula by inertia is moving in the opposite. It slides, that's why the cavity is spherical, because it slides always freely in one of those directions. And sliding results in the displacement of stereocilium, opening of potassium channels, and in this way the action potential is generated. And notice that hair cells, as you can see, in this case, hair cell has its kinocilia on the right side, but neighboring has its kinocilium on the left side. So if macula is displaced rightwards, then the first cell will be excited, but the second cell will be hyperpolarized. Not excited. In case macula moves in the opposite direction, the vice versa. This cell gets excited, this is relaxed. That's how we distinguish direction, not only the fact of movement. Okay, in general, macula helps us to perceive the vibration as well and movement along the lines, or in other words, we call this linear acceleration. So nothing's difficult. I think if you imagine once, everything will be very clear for you. Do not just confuse macula in the utricle and saccule, which is needed for linear acceleration, with the Crista ampullaris, which is inside semicircular ducts and needed to perceive rotational movements. Okay, and of course, do not confuse organ of hearing, which is in the cochlea, cochlear duct, with the organ of equilibrium, which is represented by those two parts. And at the end, several words about the embryonic development. Next slide will continue text, now just explanation. So 3 general sources for the development of the ear we have. First one is the otic placode. Otic, like for otos, ear. You're already familiar with the lens placode, with the olfactory placode, which are also derived from the ectoderm. And now the third one, the last one, there is an otic placode. It forms by invagination of the ectoderm. As a result, the vesicle is formed, and further, this vesicle is transformed into the membranous labyrinth. So, otic placode, ectoderm, results in the formation of the membranous labyrinth, internal ear. Okay, what about the middle and external ear? They are formed by 2 different sources, and let's remember the embryonic development of our pharynx. And you remember that in the ectoderm of the pharyngeal region, there are invaginations which are called pharyngeal clefts. Clefts are formed by ectoderm. Then the pharyngeal pouches are formed by the invagination of the endoderm. And as you can see, that pharyngeal pouch made up of ectoderm, it meets the pharyngeal groove, as it is written here, of the ectoderm, and on the border they are going to form the tympanic membrane. So tympanic membrane is lined by ectodermal epithelium on one side from the external acoustic meatus and by endodermal epithelium from the side of the middle ear cavity. And why we call this pharyngeal and sometimes bronchial it is also called. In fishes, usually they fuse one with another and the gill is formed, and in this way they pass the water through the mouth, through those gills, oxygenating their blood. But in humans, gills do not form. But evolutionary, our tympanic membrane is almost like a gill. It is formed by the fusion of the endoderm and the ectoderm, but they do not rupture. So there is no direct communication. And notice that the whole middle ear cavity, as well as auditory tube or eustachian tube, they are formed by the endoderm. So eustachian tube communicates middle ear with the nasopharynx, because in the embryonic development they grow from the nasopharynx, so they are formed like outpouching of our nasopharynx. That's why they keep this communication, and we should take it into account. So the derivation of this epithelial lining is the same as the nasopharynx, endoderm. And eventually, what about the ear ossicles? Ossicles are formed by pharyngeal grooves, and here we can see the pharyngeal arches, pharyngeal arches, the first and the second one. The first giving rise to first two, incus and malleus, and the stapes is formed from the second arch. So here the mesenchymal tissue forms three ossicles, and notice they are formed outside the middle ear cavity, and only with the time they get invaded, and that's why they are covered by its epithelium from all the sides, and also they are fixed by the ligaments to the walls, because eventually they get invaded. That's why they're fixed to the walls. Okay, so let's summarize the information. So you can see here bluish one, green one, and pinkish one, three different sources. Green one is the otic placote, gives rise to the membranous labyrinth of the internal ear. Then pink one, this is the pharyngeal groove, this is the ectoderm, and it gives rise to the external acoustic meatus and the outer lining of the tympanic membrane. Then blue one, this is the first pharyngeal pouch, which is of endodermal origin, and it gives rise to the auditory tube, to the middle ear cavity, and the inner lining of the tympanic membrane. And eventually, three ossicles, they derive from the mesenchymal tissue of the first and second pharyngeal arch. This information is summarized here in this slide, and I think that there are many more details, but at least this information you should understand, because really ear, it has a little bit complicated development, and it is regulated by the neural crest particularly. That's why different problems with the external ear auricle, they are usually associated with multiple congenital abnormalities. So we should pay attention and comprehend that. Okay, thank you for the attention. Thank you for subscribing to our channels. For all the questions and comments, I'm very glad to receive them all. Okay, thank you and good luck.